The blood transports oxygen and carbon dioxide between the lungs and other tissues throughout the body. These gases are carried in several different forms, dissolved in the plasma, chemically combined with hemoglobin, or converted into a different molecule. Your goals for learning are to explore how oxygen is transported in the blood, to explore how carbon dioxide is transported in the blood, to understand the effect of variables such as PO2 and PCO2 on oxygen and carbon dioxide transport. Here's what you need to know: the definition of partial pressure, the processes of external and internal respiration, the definition of a buffer. To see definitions of terms, click the bold red words. If you use a link button, you can return to the page you started from by clicking the Return button. Let's begin by looking at the transport of oxygen during external respiration. With its low solubility, only approximately 1.5% of the oxygen is transported dissolved in plasma. The remaining 98.5% diffuses into red blood cells and chemically combines with hemoglobin. Click a red blood cell to look at a hemoglobin molecule. Within each red blood cell, there are approximately 250 million hemoglobin molecules. Each hemoglobin molecule consists of a globin portion composed of 4 polypeptide chains. It also consists of 4 iron-containing pigments called heme groups. Each hemoglobin molecule can transport up to 4 oxygen molecules because each iron atom can bind 1 oxygen molecule. When 4 oxygen molecules are bound to hemoglobin, it is 100% saturated. When there are fewer, it is partially saturated. Oxygen binding occurs in response to the high partial pressure of oxygen in the lungs. Click the hemoglobin molecule to see oxygen loading. When hemoglobin binds with oxygen, it is called OXYHEMOGLOBIN. Did you notice that once an oxygen binds to hemoglobin, the other oxygen molecules bind more readily? This is called cooperative binding. Hemoglobin's affinity for oxygen increases as its saturation increases. Click the hemoglobin molecule again to observe the affinity change. The formation of oxyhemoglobin occurs as a reversible reaction and is written as in this chemical equation. In reversible reactions, the direction depends on the quantity of products and reactants present. In the lungs, where the partial pressure of oxygen is high, the reaction proceeds to the right, forming oxyhemoglobin. In organs throughout the body where the partial pressure of oxygen is low, the reaction reverses, proceeding to the left. Oxyhemoglobin releases oxygen, forming DEOXYHEMOGLOBIN, which is also called reduced hemoglobin. Click the hemoglobin molecule to see oxygen unloading. Notice that the affinity of hemoglobin for oxygen decreases as its saturation decreases. Click the hemoglobin molecule again to observe the affinity changes during oxygen loading and unloading. The degree of hemoglobin saturation is determined by the partial pressure of oxygen, which varies in different organs throughout the body. When these values are graphed, they produce the oxygen-hemoglobin dissociation curve. Notice that the axes on the graph are partial pressure of oxygen and percent saturation of hemoglobin. Click the alveoli to see hemoglobin saturation in the lungs. In the lungs, the partial pressure of oxygen is approximately 100 millimeters of mercury. At this partial pressure, hemoglobin has a high affinity for oxygen and is 98% saturated. Now click the tissues. In the tissues of other organs, a typical partial pressure of oxygen is 40 millimeters of mercury. Here, hemoglobin has a lower affinity for oxygen and releases some, but not all, of its oxygen to the tissues. When hemoglobin leaves the tissues, it is still 75% saturated. Notice that the oxygen-hemoglobin dissociation curve is an S-shaped curve with a nearly flat slope at high PO2s and a steep slope at low PO2s. We will explore the significance of the slope differences over the next several pages. Let's look more closely at the flat region of the graph between 80 and 100 millimeters of mercury. In the lungs at sea level, a typical PO2 is 100 millimeters of mercury. At this PO2, hemoglobin is 98% saturated. In the lungs of a hiker at higher elevations or a person with particular cardiopulmonary diseases, the PO2 may be 80 millimeters of mercury. At this PO2, hemoglobin is 95% saturated. Notice that even though the PO2 differs by 20 millimeters of mercury, there is almost no difference in hemoglobin saturation. This means that although the PO2 in the lungs may decline below typical sea level values, hemoglobin still has a high affinity for oxygen and remains almost fully saturated. Now let's look at the steep region of the graph between 20 and 40 millimeters of mercury. A PO2 of 40 millimeters of mercury is typical in resting organs. At 40 millimeters of mercury, hemoglobin has a lower affinity for oxygen and is 75% saturated. In vigorously contracting muscles, would you expect the PO2 to be lower or higher than in resting muscles? Click the correct answer. Correct. An actively contracting muscle uses more oxygen, so it has a lower PO2 than a resting muscle, typically 20 millimeters of mercury. At this PO2, hemoglobin is only 35% saturated. As the PO2 decreases, hemoglobin releases much more oxygen to the tissues. This allows the body to closely match oxygen unloading by hemoglobin to oxygen utilization by the tissues. Let's look at other factors which alter hemoglobin saturation. 4 other factors are pH, temperature, PCO2, and a molecule called BPG, or 2,3-biphosphoglycerate. All of these factors, together or individually, play a role during exercise. During vigorous exercise, contracting muscles produce more metabolic acids, such as lactic acid, which lower the pH, more heat, and more carbon dioxide. In addition, higher temperature and lower PO2 increase the production of BPG by the red blood cells. These conditions decrease hemoglobin's affinity for oxygen, releasing more oxygen to the active muscle cells. As an example, let's look at a graph of the effect of decreased pH on hemoglobin saturation. Click the weightlifter to see this new graph. Let's look at a typical PO2 of active muscle, 20 millimeters of mercury. At a normal blood pH, hemoglobin releases 65% of its oxygen to the muscle cells and remains 35% saturated. However, as the muscle cells release lactic acid, the pH decreases and hemoglobin releases 75% of its oxygen, leaving it 25% saturated. When pH decreases, the curve shifts to the right. This represents increased oxygen unloading compared to normal blood pH. A similar shift to the right occurs in response to increased temperature, PCO2, or BPG. If body tissues are chilled, their hemoglobin saturation changes. Which of the following graphs depicts the change in the oxygen-hemoglobin dissociation curve in response to decreased temperature? For reference, the normal curve is given in gray. Click the correct graph. That's correct. At decreased temperatures, hemoglobin's affinity for oxygen is higher, so hemoglobin releases less oxygen to less active tissues. Represented graphically, the curve shifts to the left. A similar shift to the left occurs in response to increased pH, decreased PCO2, and decreased BPG. Now let's look at carbon dioxide transport. Recall that carbon dioxide is produced by cells throughout the body. It diffuses out of the cells and into the systemic capillaries, where approximately 7% is transported dissolved in plasma. The remaining carbon dioxide diffuses into the red blood cells. Within the red blood cells, approximately 23% chemically combines with hemoglobin, and 70% is converted to bicarbonate ions, which are then transported in the plasma. Click the red blood cell to further explore carbon dioxide transport. Of the total carbon dioxide in the blood, 23% binds to the globin portion of the hemoglobin molecule to form CARBAMINOHEMOGLOBIN, as written in this equation. CARBAMINOHEMOGLOBIN forms in regions of high PCO2 as blood flows through the systemic capillaries in the tissues. The formation of carbaminohemoglobin is reversible. In the lungs, which have a lower PCO2, carbon dioxide dissociates from carbaminohemoglobin, diffuses into the alveoli, and is exhaled. Of the total carbon dioxide in the blood, 70% is converted into bicarbonate ions within the red blood cells in a sequence of reversible reactions. The bicarbonate ions then enter the plasma. Click the red blood cell to follow the reactions. In regions with high PCO2, carbon dioxide enters the red blood cell and combines with water to form carbonic acid. This reaction is catalyzed by the enzyme carbonic anhydrase. The same reaction occurs in the plasma, but without the enzyme, it is very slow. Click the carbonic acid molecule to continue the reaction. Carbonic acid dissociates into hydrogen ions and bicarbonate ions. The hydrogen ions produced in this reaction are buffered by binding to hemoglobin. This is written as HHB. In order to maintain electrical neutrality, bicarbonate ions diffuse out of the red blood cell and chloride ions diffuse in. This is called the chloride shift. Within the plasma, bicarbonate ions act as a buffer and play an important role in blood pH control. To see the reactions as a continuous process, click the red blood cell. In the lungs, carbon dioxide diffuses out of the plasma and into the alveoli. This lowers the PCO2 in the blood, causing the chemical reactions to reverse and proceed to the left. Click the red blood cell to follow the unloading of carbon dioxide. In the lungs, the bicarbonate ions diffuse back into the red blood cell, and the chloride ions diffuse out of the red blood cell. Recall that this is called the chloride shift. The hydrogen ions are released from hemoglobin and combine with the bicarbonate ion to form carbonic acid. Click the carbonic acid molecule to continue. Carbonic acid breaks down into carbon dioxide and water. This reverse reaction is also catalyzed by the enzyme carbonic anhydrase. To see the reactions as a continuous process, click the red blood cell. We will now summarize the ways in which oxygen and carbon dioxide are transported. Let's start with external respiration in the lungs. Although we will look at the processes step by step, they actually occur simultaneously. Remember, gases always follow their partial pressure gradients. Click the red blood cell to start. During external respiration, a small amount of oxygen remains dissolved in the plasma. However, the majority continues into the red blood cells, where it combines with deoxyhemoglobin to form oxyhemoglobin, releasing a hydrogen ion. When hemoglobin is saturated with oxygen, its affinity for carbon dioxide decreases. Any carbon dioxide combined with hemoglobin dissociates and diffuses out of the red blood cell through the plasma and into the alveoli. In other words, oxygen loading facilitates carbon dioxide unloading from the hemoglobin. This interaction is called the Haldane effect. Now click the hydrogen ion to see carbonic acid formation. The hydrogen ion released from the hemoglobin combines with a bicarbonate ion, which diffuses into the red blood cell from the plasma in exchange for a chloride ion. Recall that this exchange is the chloride shift. The reaction between hydrogen and bicarbonate ions form carbonic acid. Click the carbonic acid molecule to see carbon dioxide formation. Carbonic acid then breaks down into water and carbon dioxide, catalyzed by the enzyme carbonic anhydrase. The water produced by this reaction may leave the red blood cell or remain as part of the cytoplasm. The carbon dioxide diffuses out of the red blood cell into the plasma and then into the alveoli. The small amount of carbon dioxide transported in the plasma diffuses into the alveoli. To see the reactions again, click the red blood cell. Now let's summarize internal respiration in the tissues. Click the red blood cell to start. During internal respiration, a small amount of carbon dioxide remains dissolved in the plasma, but most continues into the red blood cells, where much of it combines with water to form carbonic acid. The carbonic acid then dissociates into hydrogen and bicarbonate ions. Click the bicarbonate ion to continue the reactions. During the chloride shift, bicarbonate ions diffuse out of the red blood cell in exchange for chloride ions. Bicarbonate ions act as buffers within the plasma, controlling blood pH. Within the red blood cell, hydrogen ions are buffered by hemoglobin. When hemoglobin binds hydrogen ions, it has a lower affinity for oxygen. As a result, oxygen dissociates from hemoglobin, diffuses out of the red blood cell and into the tissues. The interaction between hydrogen ion binding and hemoglobin's affinity for oxygen is called the Bohr effect. By forming hydrogen ions, carbon dioxide loading facilitates oxygen unloading. The small amount of oxygen transported in the dissolved state also diffuses out of the plasma and into the tissue cells. Click the hemoglobin molecule to form CARBAMINOHEMOGLOBIN. DEOXYHEMOGLOBIN has a high affinity for carbon dioxide, so carbon dioxide binds to hemoglobin to form CARBAMINOHEMOGLOBIN. To see the reactions again, click the red blood cell. Here's a summary of what we've covered. O2 is transported in two ways: dissolved in plasma and bound to hemoglobin as oxyhemoglobin. The O2 saturation of hemoglobin is affected by PO2, pH, temperature, PCO2, and BPG. CO2 is transported in three ways: dissolved in plasma, bound to hemoglobin as carbaminohemoglobin, and converted to bicarbonate ions. Oxygen loading facilitates carbon dioxide unloading from hemoglobin. This is known as the Haldane effect. Carbon dioxide loading through the formation of hydrogen ions facilitates oxygen unloading from hemoglobin. The effect of decreased pH on oxygen unloading is known as the Bohr effect.